This week on Surfing the NASH Tsunami.
And that'll wrap up the first day. What a fun-filled day that is. That covers the gamut from clinical trials to platform trials to challenges related to clinical trial development and all the way down to artificial intelligence.
Join hepatology researcher and key opinion leader Dr. Stephen Harrison, liver wellness advocate Louise Campbell, pricing and forecasting guru Roger Green, and this week's guest, hepatology researcher and clinician Ian Rowe, as they preview next month's NASH-TAG 2021 meeting from their own unique perspectives on the Surfing the NASH Tsunami podcast.
For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 2, Episode 8 of Surfing the NASH Tsunami starts now. Good news, everybody. Stephen's come out of the cold, has his electricity and his heat back, and is joining us to lead today's discussion. He's got a lot of work left to do because San Antonio is still a problem. He may talk about Starbucks in a minute, but he's back with us today. And more good news, our friend Ian Rowe is with us today as well for the first time this year. Ian, how are you?
I'm really good, thanks, Roger. It's a pleasure to be back on the podcast.
Glad to have you with us. Consistent with the format we've started, headlines about the podcast. Another great week in terms of listeners, and an exciting episode for next week. We'll talk about that after today's dialogue. And this is Monday, if anybody tried to get in this morning. Buzzsprout was subject to a DDoS attack on Saturday, and they spent 48 hours intermittently on and off working to get the site straightened out. We're told everything is fixed now, and I saw people started successfully downloading again a couple hours ago. Maybe that's the end of that. If they give us more news, I'll give you a story next week because it's an interesting thing. This week, we're previewing the 2021 edition of NASH-TAG, which runs March 10th to 12th in Park City, Utah. I'll talk a little bit about the importance of NASH-TAG to me after intros, and then Stephen will walk us through a review of the program with questions from the rest of us. Let's get started. One good thing that happened in the past week, professional or personal.
This is Stephen. So I would say the best thing that happened in the past week was that we survived. So as many of you know, Texas was the center of an Arctic vortex blast that rendered the entire state rather helpless. And we have affectionately termed it Snowvid, Snowpocalypse. You know, you come up with the word that involves cold and snow. This idea of rotating power outages to maintain the grid, I think was an interesting one, but it was a disaster. So, you know, we would have power for 90 seconds and it would be off for the next 12 hours. And I've never craved coffee as much as I have during the past week. And so I have a Keurig at home, and I would have it all set to go with the water in the machine and my little pod in there. And literally when the electricity came on, it was a sprint faster than Usain Bolt to the Keurig to turn it on and see if it could make a cup of coffee before we lost electricity again. And that was only complicated by the fact that we had no water. And then when we had water, we had to boil the water. But suffice it to say, there were a lot of people that were a lot worse off than me. We just snuggled in the car under blankets and heat until we ran out of gas, and then we just kind of snuggled next to a log fireplace in the living room that was gas. So we didn't die of carbon monoxide poisoning. We're here to live and to tell the story, and we're slowly getting back to normal. The comment that Roger made about Starbucks was you can use it as a marker of whether or not your society is back to normal, is whether or not Starbucks is open. And it had been closed since last Saturday, and as of this morning, it's still not back open yet. So we're all anxious for that to reopen and as a sign of returning to normalcy. But, you know, what a week.
Well, welcome back, and glad you made it through. We've had harrowing storms up here, but we've lost power for 10 days at a time. It's pretty awful, and we know how that works.
Oh, I can go next. It's half term here, so we were able to get the youngest out of school or away from home learning in a different location back to home. So that was quite nice for the weekend.
Yeah, and I think this evening's brought the news that we're finally looking towards getting our children back into school full-time with all of their friends in the next 2 weeks, which I think will be a welcome relief for both our children, but also for many colleagues, parents and other kids who really need to get back into some degree of normal life and back learning in a proper environment.
Generally, the good news in the States is that the new infection rate is dropping so quickly, and that's even in advance of vaccines coming in as strongly as they will. There are reasons to wonder whether that will hold up, variants and things like that, but it feels a little less scary, at least for now, and I think that's a good thing for everybody, since people have been quite stressed about this, even if they haven't had challenges around weather. We've now gotten to the end of our SNOVID openings. They were all COVID or snow, or combination of the two. And with that, on to NASH-TAG. NASH-TAG has a warm spot in my heart. It's where I first encountered the NASH community. I was invited to come speak about commercial issues in 19 and 20. Met Stephen, had seminal conversations with people that led me to really appreciate fatty liver disease and the challenges it faced. And forget about me, what NASH-TAG has really been is the place where a select group of NASH stars, drug developers, key opinion leaders, investors, C-suite execs, meet to discuss the science of fatty liver, review some of the exciting drugs in development, and debate, literally debate with pro and con sides, key issues in liver drug development. Last year, NASH-TAG was the first place, I believe, that Stephen shared his data publicly on intra and inter-reader reliability of histopathology, which caused a fairly rapid shift in many people's thinking about exactly how good the gold standard is. 2 years ago, a panelist suggested rethinking the value of the drugs and pricing models for the class, and early-stage companies started revamping their strategies and projections. Because it's small and because it's select, when somebody says something that puts on light bulbs, people take action, which makes it really a unique meeting in those that I've experienced in my life in pharmaceuticals and healthcare. And this year it'll be a hybrid. First in-person meeting in 14 months and coupled with online capabilities. 4 co-directors, Vlad Ratsiu, who we see on the podcast from time to time, and then Michael Charlton and Rohit Lumba and Stephen. So Stephen, at this point, I'm going to turn it over to you. I want to ask you if there's anything that you think makes this year particularly special. And then after that, if you can walk us through the day 1 and day 2 sessions, and then we'll come back to you with questions. That would be great.
Yeah, thanks, Roger. So if I'm remembering right, this is our 5th NASH-TAG. Time flies. The genesis of this meeting was all about science. The 4 of us sat down and said, how can we have a meeting completely focused on NASH and drug development that is so focused in thought that everybody that shows up is tuned in to this one area of liver disease, and to not be distracted by everything else that's going on around them. You know, I love AASLD and I love EASL and I go every year, but it's crazy. You know, everybody's running from session to session, from hotel room to hotel room to ballroom to ballroom to meeting room, trying to catch up with friends in the evening, multiple different meetings with multiple different companies and other thought leaders. And you're trying to work with your fellows and with your colleagues. You're trying to mentor. You're showing up at AASLD governance meetings, NASH-SIG meetings. You're giving lectures. You're trying to learn some areas of the disease state that maybe you don't know. So you're attending cholestatic meetings and liver transplant meetings. And it's a ton of learning. It's phenomenal, but it's not focused on NASH. NASH is part of it, but it's just blended in with everything else. So we thought, what would it be like to pull together a meeting that's 2 days long in an environment where people can come and relax, let their hair down, and just be open and engaged with the challenges we're faced in drug development in the field of NASH. And it's broadened a little bit to include non-invasive testing, but we also include preclinical data as well as clinical data. And it's been well received, I would say. We've continued to max out our venue. We didn't want to expand it because we want to keep it intimate and engaging. So we've limited participation to about 230 people each year because that's the maximum we can have in the venue that we're at. And then came COVID in 2020. Fortunately, our meeting was in January, so we were able to have an in-person meeting last year. And as you know, every other in-person meeting has canceled and remains canceled. Our meeting was supposed to take place in January of this year. We postponed it to March 12th and 13th of this year because we wanted to see if buying an extra 2 months would give time for the disease to quiet down and people to get vaccinated. I think we've done that to a partial extent. And as a result, we've made it a hybrid meeting. And so this is the first time since our last NASH-TAG that people will have the ability to show up in person. I don't know how many spots are still left if you're interested in coming in person, but when I say hybrid, there's also a real-time simulcast virtually. So if you can't attend in person, you can still be a part of it virtually as well. So we're going to try out the hybrid part of this, but I will be there in person as well as Mike Charlton and Rohit Loomba. I don't think Vlad can make it. That's still a bit up in the air as to whether or not he can leave France and come in without having to quarantine for 14 days or something. So that's still to be determined. But that's probably the most exciting thing I'm looking forward to is actually seeing my colleagues and friends in person and being able to continue the dialogue of drug development, because paradoxically, this disease did not slow down in 2020, and neither did drug development or non-invasive testing for the disease. So there's a literal ton of data to discuss that hasn't been debated in open forum yet. So if you think about the 9 press releases that came out last year with drugs related to NASH, 8 of which were positive and only 1, elafibranor, that was negative, that hasn't been debated in open forum. We presented them at EASL and AASLD in a virtual format where there was a few Q&As, a few questions that were asked and answers given, but there wasn't that chance for just intimate dialogue about the data, both good and bad. And that's one of the highlights of the first day of the meeting. So let me jump right in and kind of go through what we're going to do on Friday, March the 12th. So the meeting starts early, like it always does, at 7:30. Mike Charlton will give his seminal address on the year 2020 in review. And let me tell you, I've been to a lot of lectures in my career. This one that Mike gives every year on the year in review for NASH is absolutely phenomenal. Amazing. He puts careful thought into this. It takes him about 8 months to make this lecture. It seems like he starts on it in the summertime and is very creative at interpreting the data that's been presented on a global scale. So that's really a can't miss opening salvo, if you will. Then historically, what we do is we get into clinical trial results with histology data. So what we're going to do here is a format called presentation with rebuttal. So we have a presenter who presents the clinical trial data, and then another presenter right behind him will come up and rebut the data. It doesn't necessarily mean that rebuttal is completely negative. It just means that it's looked at through a different lens. And so the first presentation is on tropifexor. As you know, Arun Sanyal presented this at AASLD. He will present it again at NASH-TAG, and then Vlad Ratsiu will rebut the data. There will be an open mic discussion. This is what makes it so fun, is people get up and ask questions to both of those presenters about the data, and Mary Ranella and Rohit Loomba will moderate the session. So it'll be a fantastic discussion about FXRs in general and tropifexor specifically. That's immediately followed by lanifibranor and the data from Inventiva. Professor Abdumalik will present the data, and Brent Tetry will rebut that, again with open mic discussion. And then finally, semaglutide will be presented by Phil Newsom. You know that paper has now been published in New England Journal of Medicine, and Mazen Nareedin will rebut that with open mic discussion. That'll conclude the first session. Around 10 o'clock, we'll start session 2, and this is really not a deep dive, but just a closer look at some of the early phase 2A trials. Rohit Loomba will present the FGF2189 bio phase 2A data. I will present the Akero efruxifermin data, and Rohit Loomba will present the FASN inhibitor Sagimet data, and we'll have a round of discussion on those 3 trials. Adrian DiBascelli and Manu Chakravarthi will moderate that session. And then we immediately roll into a mini debate. This is kind of a novel thing. We haven't done this at NASH-TAG before, but we're going to look at beyond success stories. Why do so many NASH trials fail? And we're going to first talk the non-cirrhotic trials, and Vlad Ratsiu will present that, and then we'll talk the cirrhotic trials. Mary Ranella will present that. I think that's going to be a fantastic opportunity to listen to these guys talk through why some of these trials, why so many of these trials are failing. And what I'm talking about failing, I'm not talking about screen fail rates and that sort of thing. I'm talking about drugs that didn't make it across the finish line. It seems like the graveyard is filling up a bit. And we're going to talk through lessons learned that we can apply to future trials to prevent that from happening. And then there's a discussion on rapid fire on promising data. Now, this is interesting. Mike Charlton, Rohit Lumba, and Naeem Al-Khoury are going to just go round table and talk about promising data that's coming out, just a rapid fire sequence, and then we'll have time to engage with that as well. And then in the evening, as you guys know, there's a break between 12 o'clock and 4 o'clock where I call it interactive dialogue on NASH topics. As you know, the setting is Deer Valley. This time of year, there's a white powdery substance on the side of the hill called snow, and some people like to engage in some interactive dialogue on NASH topics while riding up the mountain on chairlifts and then coming down. And so that'll continue for 4 hours, and then we regroup at 4 o'clock and introduce this concept of platform trials. This is going to be given by Senior Director of Clinical Development and Cardiovascular Metabolism from Janssen Pharmaceuticals, Nicholas De Prospero. And I think this is an interesting, novel way to approach NASH. It's an innovative trial design. And really the concept is it fact to it? Is there legs to this idea? Or is it just a pie in the sky mentality? And then Vlad Ratziu will present again on specific challenges and opportunities for NASH clinical trials. And we'll move into a panel discussion where we debate all of those issues. So we'll debate platform trials, we'll debate specific challenges and opportunities for NASH trials, and that will be with Kitty Yale from Akero, with Xiao Liu from NGM, Hank Mansbach from 89 Bio, Vlad Ratziu, and then Nicholas De Prospero from Janssen. And then I'm going to come on and talk about seladelpar's FDA hold and release. What happened with that? What did we learn? It's really a deep dive into understanding histopathology relative to NASH, and maybe some heretofore unreported or unrecognized lesions in NASH that are actually maybe more common than we previously thought. And then we move into artificial intelligence. Dr. Quentin Anstey will present on automated histological analysis. Seeing Beyond the Pathologist's Eye is the title of his lecture. And then we'll pivot to 2 company lectures, one from PathAI on what does it take for automated histologic analysis prime time? What does it take to be accepted by the FDA? Are we there yet? How soon will we get there? And then Dean Tai from HistoIndex will talk about breaking down cirrhotic lesions and implications for drug development in that arena. We'll have a great panel discussion that is led by myself and Dave Kleiner, who is the head of histopathology and autopsy pathology at the NIH, and Quentin Anstey from Newcastle. And that'll wrap up the first day. What a fun-filled day that is, that covers the gamut from clinical trials to platform trials to challenges related to clinical trial development and all the way down to artificial intelligence. So maybe I'll stop there and take a breath and see what you guys think. Wow,
all sounds very, very exciting, I have to say. Very scientific. And it'd be great to have some of you guys there. We certainly can't travel from the UK at that time, but some really, really interesting topics you described there, and I'm certainly looking forward to seeing what Phil Newsome and the guys present on their session particularly.
Yeah, I think more so than previous years where some of this just gets recycled and we have time for a little bit of a deeper dive than what occurred at AA or EASL. Here, we all get the sense that a lot of this data hasn't really been discussed. The meat hasn't been pulled off the bone, and a deep dive done into what was presented and questions that still remain. So a lot of the first half of this day is going to be just taking the FXR field, the PPAR, pan-PPAR field, the GLP-1, the FGF21, the FASN inhibitor, and really breaking those down and beginning to understand what positive, and if there is negative, what that negative looks like relative to each of these. It used to be we just talked about FXRs. Now FXRs are still in play, but they're just one of many. We're having a comeback in the PPAR class, right? There was pioglitazone, and then there was elafibranor, and now there's lanifibranor. And so positive data coming out of there. We're still struggling a bit with the weight gain issue, and can we get past that? Semaglutide, we're going to do a deep dive into that. Maybe we'll have an open mic discussion that will reverberate a little bit around why there was a lack of fibrosis benefit with semaglutide. I hope there is a robust discussion on that topic. My own belief is I think there's some something in the way the biopsies are interpreted, and maybe artificial intelligence would have a role to play here in fully quantitatively assessing collagen from that trial. I could go on, but I think you get the point. We're beginning to do maybe a deep dive and run to ground some of these unanswered questions that still remain with these different classes of medication.
Fantastic. Do me a favor, Stephen, before we keep going. For those who are listening who might not understand the concept, because lots of different kinds of people listen to this, could you please explain what a platform trial is and how it differs from what folks are accustomed to?
Well, it's a bit of a new concept that I don't fully understand all the nuances of myself, Roger, but it's basically just a different way. It's decentralizing clinical trials and potentially can be leveraged across multiple different drug platforms. So without diving into it too much more, I'd say stay tuned.
I think that's going to be a really interesting discussion about platform trials and, how you can integrate different drugs and different arms into studies. And particularly as combination therapies begin to be fully evaluated, it provides an opportunity to do that in a way that you can get meaningful data relatively quickly. But it's very much an efficacy experiment, if you like. And I think one of the big challenges that, in some ways, COVID has shown this in the way that the recovery trial's been done in the UK, where we've tested the effectiveness of a series of agents against COVID and shown robust effects, but in very large populations. And that platform approach, I think, is likely to be very useful in defining whether a drug is going to show some efficacy, but whether those drugs will be effective is always going to take much bigger studies. And I think the experience that you'll get from a cardiovascular researcher in that field will be, well, at least I hope it'll be enlightening, and should probably provoke a really strong discussion about understanding efficacy on one hand and effectiveness on the other, particularly as we begin to get drugs through that look like they're really going to have a big effect on rates of disease progression.
First of all, sounds like a great day. One of the things I've learned being at NASH-TAG in 19 and 20, and I can't go this year because I'm not going to be vaccinated in time, is that knowledge builds on itself over the life of the conference. First, in 2020, you're right, there was FXR to talk about. And the big insights were around testing, and I think largely started with your paper on inter-reader reliability and what did that really mean for the value of histopathology as gold standard. But here it sounds like you're going to be laying up 5 different classes of drugs in development, each of which is at least somewhat promising and has at least one flagship agent doing well, and asking, okay, how do we put these pieces together? What makes sense? What will get used how? It just sounds like an extraordinary look into the future.
Well, that's the hope. It's encouraging to see positive data coming out, not just on non-invasive tests, but on liver biopsy data. But I also think what this meeting gives us an opportunity to do is really begin to harmonize our thoughts on the way forward. Can we gain consensus around how artificial intelligence should be incorporated? Can we gain consensus around how paired liver biopsies should be read by pathologists based on the FDA guidance that recently came out that said we're not beholden to any one way of doing it. We're willing to listen to novel ways of interpreting histopathology where we can minimize variability, minimize intra-observer variability, minimize placebo response rates. And so I think that's where I've seen NASH-TAG be really beneficial, is being able to harmonize thoughts on lots of different topics. And to your point, there's a lot of positive data that's being presented. So what does the future look like relative to all of that? Do we have one size fits all? What drugs do we begin to co-formulate and study together? Do we do induction therapy followed by maintenance therapy? All of these discussions will be had, whether they're going to be had in public forum or they'll be had in sidebar conversations at the meeting. Rest assured they will be had. And I think this is where, we can all read the literature, but when you're siloed and you're stovepiped in a Zoom meeting, you're limited in your ability to think outside the box. And I know in my own career, some of the best thoughts I've had were thoughts that were triggered by a sidebar conversation with one of my colleagues over a soda, a beer, you name it, over dinner. And that triggered a thought process and a discussion that subsequently ensued that gave me something to think about when I came home. And I applied that in the next study that I did, or in the next review that I wrote, or in the way that I presented things. And I think that's really what what happens at a meeting like this. You take all the data and you synthesize it, you boil it down, and you walk away with, okay, there's the what, we talk about the data, then there's so what, we debate it and throw it back and forth and chew on it collectively, and then there's the now what are we walking away from and doing as we leave the meeting? And I think that what, so what, now what mentality is really what's made NASH-TAG a success.
Question, Stephen. The many debates, the beyond success stories, so why do many trials fail? I appreciate that NASH-TAG is all about the science, but the patient is beyond science, and the patient or the subject is a massive confounder in why clinical trials fail. But there's no person, there's no subject who's lived a trial life talking about what they found difficult, what they found hard, what they changed that was just something that could change an outcome. And we know you don't have to change a lot. You only have to change the quality of your food for some of these subjects to significantly change internal structure. And I think, will that be looked at? Because what I'm very used to in the medical world is when scientists get together and are very science-orientated, sometimes we lose the humanistic side, the bit that we can't legislate for because we like to put things in boxes. Is there an opportunity within that forum that's going to be presented from the opposite side of why trials fail patients?
That's a great comment and a great observation, Louise. We have historically had patient advocacy groups represented at NASH-TAG, not necessarily on the podium, but in the audience. And we've always encouraged interactive open mic dialogue with the advocates, the patient advocates that are there. So I don't know exactly, I haven't seen Dr. Ratsiu's lecture on exactly what he's tackling in his title, Specific Challenges and Opportunities for NASH Trials, but I suspect that will come up, and if not, maybe virtually you can raise your hand and ask that question, because I think it's an incredibly important one to include.
Yeah, I know. And I know Donna's going, so I think she said she was going to try and be there in person, wasn't she? So GLI will definitely be represented, which is good news.
And I think Wayne usually shows up too from the Fatty Liver Foundation.
Yeah. So Stephen, you mentioned virtual questions. What opportunities will people who are not in the room have to participate in the meeting?
My anticipation and maybe expectation is that they will have the ability, just like on any typical Zoom platform, to raise their hand or type in a question. Moderators should and would be expected to address some of those questions. So that's how we're going to do it, and unless I'm told otherwise, that's the way we'll plan on doing it.
I'd love to leave enough time for day 2 because it's a couple different directions and really fascinating. So unless someone has something else they want to ask specifically about the phase 1 agenda, Stephen, why don't you come walk us through phase 2?
Day 2 starts Saturday, March 13th at 7:30. We're going to talk about what would NASH trials without histology look like. I'm going to moderate that with Vlad Ratsiu, and Mary Ranella will kick us off with serum biomarkers and prediction of outcomes. So remember, there are 3 different contexts of use for a biomarker in the field of NASH. One is diagnosing the at-risk NASH patient. Number 2, measuring therapeutic efficacy, right? You put them on drug. Are they responding? Can I stop them? Do I need to keep treating them? What can I tell my patient the response is likely to be if we did a liver biopsy? And then finally, determining long-term outcome data. So Mary is going to walk us through what that looks like. Then immediately following, Professor Lumba is an expert in imaging biomarkers. He's going to show us what imaging biomarkers and prediction of histologic response and fibrosis change in outcomes have looked like over the past year. He's presented a lot of data on MRI, PDFF, and even MR elastography. So we'll hear from him on that. And then an interesting conversation will ensue from a guy I don't know well, but Mike Charlton recommended him. And Ian, I don't know if you know him. His name is David Pinato. He's at Imperial College in London. He's going to give a talk on precision medicine, incorporating biomarkers into clinical trials. Don't know a whole lot about that, but Michael says he's phenomenal, and we are in for a treat on that lecture. That will be followed by Phil Newsom talking about ready to select patients with active NASH and fibrosis without biopsy. In other words, can we use our non-invasive testing strategy to identify the at-risk NASH patient with fibrosis and actually not do a liver biopsy, but include them in our phase 2 and phase 3 trials? Now, the FDA is still advocating liver biopsy here, but what would it look like if we didn't have liver biopsy? Because I think that's where we're eventually headed. And that's actually the title of the next talk, which is What Would Nash Trials Without Biopsy Look Like? And Vlad Ratsiu will give that talk. And then every one of them will come up for a panel discussion. It'll be myself, Vlad, Sunil Hasmani from GenFit, who heads up the NIS4 biomarker development program, Celine Fournier from EchoSense, who's the chief medical officer there, and hopefully Rob Myers from Gilead, although I think we're still waiting to see if he can join us. And that'll be the first session. So a really robust dialogue, right after breakfast, on what would NASH trials look like without histology. Then we're going to roll into special populations in NASH trials, something we haven't done before either. We're going to talk about NASH and HIV and other unique populations. Is this a specific unmet need? And then Quentin Anstey is going to talk about the elephant in the room, NASH and moderate alcohol consumption. There's a lot of debate out there about, are we really including alcoholics in some of our NASH trials? And is there a way to mitigate that? And what would it look like? And then we're back to our mini debates before we finish the morning session. And this is on diabetes drugs for NASH. No need to look elsewhere. In other words, is that all we need? And Sven Frank is going to take the idea of, yes, indeed, it's all we need. Arun Sanyal will come back with not so fast. And then there's a panel discussion that will ensue. And then from noon to 4, we're going to entitle this a la carte roundtable discussion related to drug development. This, again, is that opportunity to maybe get out in nature and have discussions in a roundtable format with your colleagues, maybe while you're pursuing alternative physical events. And then we'll come back at 4 o'clock in the evening and dive into basic science. And again, this is fascinating to me as a clinical scientist. I have never been brought up in the preclinical world, but I've learned a lot over the years from meetings specifically like this, where we have experts come in and really dive into some of the basic science. So Brent Tetry is going to start off with preclinical models. Lots of information. Is any of it usable in humans? I think that's an interesting topic because we know that if you don't see a lesion improve in an animal model, very unlikely to see it happen in a human. If you do see it improve in an animal model, it doesn't mean it's going to happen in a human. And the other thing is there's not really a very good animal model to replicate ballooning degeneration. And so he's going to talk through that. There will be a period for Q&A after that. And then Bernd Schnabel will come in and talk about the microbiome in NASH and implications of pathophysiology and treatment. This is an area that has been around for a while, but one that we need to really get back engaged with as we learn more and more about the microbiome. And then finally, in this session, an old icon of NASH-TAG, Scott Friedman, and I say old icon, he's literally a well-known figure in the field of fibrosis and well-respected. He's going to talk about directly targeting novel mechanisms for fibrosis, specifically TGF-beta and integrins. And then this session will finish out with advances in genetic and gene silencing in NASH by Professor Lumba. And then our last session of the meeting will be new pharmacotherapy targets. So we'll hear from William Essler on DGAT2 plus acetyl-CoA carboxylase from Pfizer. We'll hear from the president of Terns, Aaron Quirk, on SSAO and VAP1 inhibition. Mike Charlton will talk about toll-like receptor signaling inhibition. And Robert Foster is the CEO of Hepion. Will talk about CRV431, a pancyclophilin inhibitor. Altimmune, Scott Harris, the chief medical officer, will talk about GLP-1 glucagon co-agonists. And then Dr. Lumba will come up and talk about PNPLA3 silencing. And then I'll wrap up the whole meeting with a recap and future direction. And we'll adjourn the meeting and send everybody out for dinner and the next day back home. So what do you think?
I personally am trying to figure out how many fire hoses one can drink from at a time, because boy, there's a lot here.
I think it looks excellent. And I, for many reasons, I'd love to be able to go in person. I think that first session on the Saturday morning about what trials without histology would look like, and I think that'll be fascinating because in some ways that's really the sort of medium-term view to what clinical practice is going to look like and how we might select patients for treatment without biopsy. And some of the discussions that you'll need to have within that will be quite illuminating. And I do know David Pignato, and he is excellent. His background as an HCC and looking at biomarkers, particularly of checkpoint inhibitor therapy response. And you really will get an excellent lecture from him. But I think that session in general looks really terrific.
Well, that's great to know, Ian. Thank you. I'm looking forward to that talk for sure. And that whole session, I think, again, we're going to leave about 30 minutes for a panel discussion at the end of those 4-5 lectures to really dive into this a bit more and get input from hopefully the virtual crowd as well as the in-person crowd.
I was just going to say exactly what Ian said. That morning session looks very exciting, and it's certainly, as he said, the future potentially we would hope of beyond the biopsy and of clinical practice and being able to see more people, I suppose, in the real world than we currently see getting through the biopsy process. But yes, very exciting. And also the elephant in the room of moderate NASH consumption. We know that people consume a lot more alcohol than they necessarily confess to, and how much alcohol is acceptable in a fatty liver trial. But do you remove alcohol? Do you remove a high sugar drinks that can be somewhat confounding as well? So if we're going to be interesting to look at different aspects from that.
Absolutely.
I'm also fascinated by the diabetes debate. The reason being, in very, very practical terms, at least some health agencies approve drugs based in part on how compelling the need is. So the more adequate a solution you believe semaglutide is to NASH, the less urgent you might feel about approving other agents, which might have to do with approval or it might have to do with reimbursement. But one way or another, the adequacy of what's available today, I think, will bear on how aggressively we are able to move forward with novel agents, many of which I think are really exciting. So I will be intrigued to hear particularly Dr. Frank's presentation and how you make a case that goes yes on the diabetes drugs, given both the strengths and the seeming shortcomings of the semaglutide trial.
We had fun putting that one together. I think there's a lot of energy behind the GLP-1 class because, look, let's face it, weight loss is kind of key to 90% of the NASH issue that's out there. And we know there's histopathology improvement that's linked to certain percentages of weight loss. And the GLP-1s, particularly semaglutide, injectable, have shown in obesity trials a proclivity to lose weight. And I think that's really important. The flip side to that is the tolerability profile. And if you look at claims data, how many of these prescriptions actually get renewed over time? And are patients compliant with it? So does it have a role in NASH? Absolutely, I think it'll have a role in NASH. Is it everything it's cracked up to be? Well, I think time will tell, and I think this debate will be probably one of the more fascinating and potentially controversial discussions. So I'm looking forward to that because I think it's going to close out Saturday morning with a bang.
Yeah, I agree with that. I'll tell you, one of the things that grabs me as I think about it, and remembering that Phil Newsom is presenting his semaglutide paper the day before, is that you're going to have discussion and debate on this on Friday, and then as you described, all those off-to-the-side conversations, and then come back in and do this on Saturday morning. So I think there'll be a whole bunch of ferment before you even get to this. And a lot of times people consider all these issues in the balance of everything else that's been heard. I can't wait to hear that.
Yeah, I agree.
It'll be a good debate. And following the discussion we had last week, is if you start to inform patients who are diabetes now that they've got fatty liver disease, does that impact in a positive way and improve the outcome of these medications? Because, of course, they then tend to change their behaviour a little bit more than they would if they were never told that they had fatty liver disease. For me, I'd like to see us reach that point now, with cancer being the leading cause with diabetes of deaths in that population, that whole conversation being had, and we could see these medications have far more effect with the exercise and the improved diet that people may well be triggered into with a differential diagnosis of fatty liver disease and fibrosis.
Louise, here's a side thought I had, and it involves a question to you. You think about COVID. If I told you, look, you're not going to get COVID if you lose 10% of your body weight, you won't have it. Guaranteed. 99% probability you won't get COVID. Do you think that there would be a much bigger push by patients to lose 10% of their body weight? And the reason I ask that is because there's been so much press, so much in your face about COVID. Everybody knows about COVID. They know how deadly it can be. They know there's a vaccine. People are lining up like crazy to get the vaccine. We're not having to force people to go get the vaccine. I get it. There's anti-vaxxers out there, but there's a whole lot of people. Just in my own city, we run out as quick as we get it for vaccinations. Imagine if the vaccine is weight loss. And think about NASH. Most people don't know about NASH. They still don't know about NASH. They don't know the risk of NASH. They don't know that patients can decompensate and actually die from their liver disease. And if they knew they could make a huge impact on their life by just changing their lifestyle, I think more people would actually do that. Now, granted, the data is pretty clear. Getting people to lose 10% of their weight and keep it off is a unique challenge and one that not many people can do. But I just wonder, back to the question, if the cure for COVID was 10% weight loss, how many people would be trying to achieve that on a daily basis?
I think people are trying. Obviously, you know, our Prime Minister had COVID, and I'm sure Ian will confirm any of the data that I get wrong, but he typifies in his body image somebody who probably has fatty liver disease, if I had to put my cards on the table on that and guess. But we had a weight loss, an obesity campaign. 6 million people downloaded the NHS app the last time I looked to use for weight loss. We get multiple stories on the news of people who were morbidly obese with BMIs over 40 and 35 who've taken up running, who've lost the weight, who've done it because they knew that they were really severe risk. What we didn't mention was the elephant in the room is yes, you're losing body weight, but in 99% of those people, you are going to lose, it's the liver fat, it's the pancreatic fat in your diabetic population that changes that outcome most significantly. But I genuinely think that we are so stigmatized, both within healthcare, in other professions that don't look at liver disease as their primary disease, to mention liver disease because people get offended by being, oh, I can't have liver disease, I don't drink, or I don't use drugs. So by not having those conversations, we continue to stigmatize an organ that is key to everything we do. I have a friend who I do have FibroScanned who had for his size, he had relatively low liver fat. Now, he is in hospital currently with COVID, but didn't get ventilated, went to CPAP for 24 hours and came back. Knowing his internal liver fat, would have guessed that would have been the outcome with him. But those are just the odd cases. But I totally agree with you. If we started to have those discussions about what organs are involved, you would then start to see the heart websites for patient advocacy groups mentioning to their heart patients that they might have a liver problem, and particularly the diabetes sites, to say to their patient forum that, have you been checked for liver disease? Those aren't conversations that they're having on their websites looking after their patient group. And why? Because the liver remains stigmatized within all of the other areas. It's probably one of the biggest reasons it's not been classified as a non-communicable disease, despite fueling most of the big non-communicable disease problems. So people do want to do it. And if you can measure it with something like FibroScan to say that you're making that improvement, and you can do it 3 monthly, people do stay motivated, but they don't know how much improvement they've made. That's an important factor. We do have the technology, and I know the science is all about, we want a FibroScan of 8.5 and we want a cap of above 270, 280, 300, preferably for clinical trials. But I actually don't want people to be clinical trial patients. I don't want them to have enough liver fat to cause a problem. So when I look at FibroScan as a wellness tool, it is keeping people in the lower thresholds, irrespective of their overlap. If they don't get to 300, they're less likely to have a problem. So if you can keep them motivated to keep that liver fat lower, and I don't think there's simple steatosis. We'd think that steatosis does cause problems, even if it doesn't cause fibrosis. So for me, it's keeping people out of the threshold in the long term, or the majority of people out of the threshold. We will always have those who get to those thresholds. but the numbers at the moment are massive. 14.6 million people in the UK have fatty liver disease. We know that you guys estimate 100 million in America. And I do think a lot of people would be motivated if they could see their internal change. It's not what we look like on the outside, it's what we look like on the inside that in COVID really matters. So my long answer to that is yes, I do think if we were overt in discussing the internal liver weight loss, people would be well more motivated and be able to see it. And you can see that liver loss in somebody on a FibroScan in the CAP.
A couple thoughts about that. First of all, you couldn't have teed up next week's episode a lot better if you tried, because this is exactly what we're going to be talking about next week, number one. Number two, to go back to Stephen's question, Stephen, I think the answer is complicated by the behavioral issues around weight loss, which is, I think if people had that information, the percentage that would want to lose the weight would be vast. And without better psychosocial support than we give them right now, even if they had FibroScans, many would succeed, many would fail. The problem is once you know that you've got a fatty liver inside you, which is like a ticking time bomb, failure has more severe emotional consequences to it. So I think your question is great. I think if we start teaching people those things and then provide them with better psychosocial support than we've done to date, we really can have tremendous impact on that and broad impact on that. In the absence, I think we go to where we are, which is people lose weight, but most of them can't keep it off because ultimately it's not about having a different goal. It's about, the way we approach this is the goal is to lose 10% of your body fat. The way we should be approaching it is, I am a disciplined person who am careful about what I eat because of what that says about my life. And if I tell myself that's the person I am, then I will execute the behavior. And if I execute the behavior, I lose the weight. And that's how it stays off. But we don't do that right now. And we would need to be able to support that kind of thinking and that kind of change in habit to get to where we want.
I guess I just want to circle back in my own practice. If we assume COVID is taken seriously by every patient that I encounter, and I tell them there's a vaccine ready for them, you should go get it, I think a lot of them would go get the vaccine. And I think that's predicated on the fact that they know how severe COVID can be, that it's killed 500,000 Americans, more than World War I, World War II, and Vietnam combined. But what we don't tell them is how many people died from NASH-related complications, which I can guarantee you is more than 500,000 people. Right? We don't have that type of conversation. And patients don't know that data. They know fatty liver. They don't know fatty liver. But let's say they did know fatty liver. Our primary care frontline docs, unfortunately, aren't armed with data to show them or have the time to sit down with the patients and explain the potential seriousness of the consequences that can come with having fatty liver. Forget fatty liver alone. The number one killer is cardiovascular disease, right? So again, my point is, I personally think if there was disease awareness out there for our patients with fatty liver to the same degree that there's disease awareness about COVID, and the quote-unquote cure is weight loss rather than a vaccine, I think more people would do exactly what Louise said. They would download that app and they would get after it more. They may not be successful completely, but heck, 5% is better than no percent. If you look at the data presented by from the Ian Haynes data, the vast majority of NASH patients don't do any exercise. So at least maybe beginning to do something, we might see a completely different outcome relative to long-term patient benefit.
I am just going to finish on one thing on what Stephen said there, because I think he made a very, very good point in vaccination. The people who are, certainly in the UK, more resistant to vaccinations are those ethnic populations that actually are at highest risk of fatty liver disease. Asian population, our Bangladesh population, our Arabic population. And no matter whether or not it's due to suspicion, there is no suspicion with if we ask you to lose weight. If you don't want to take the vaccine, lose weight. That can protect you. So there is a good argument to exactly what you're saying, because there are the anti-vaxxers. But actually, we're just telling you to lose weight. If you don't want to have a vaccine, lose weight. It won't protect you from getting COVID, but it may well protect you from dying. So that was an excellent point you made.
And I think this has been a fascinating little discussion about what drives behaviour change and the sort of perceived risk of various illnesses, and that sort of the publicity and sort of danger sense around COVID is a potential driver. And it always reminds me of the story about the commonest time to buy earthquake insurance is just after an earthquake. And if we don't use information at the right time, the impact of what you say to the patient is likely to be lessened. And it comes back to understanding what is going to have the greatest impact for behaviour change, and whether that relates to overall risk of death, whether it relates to COVID, whether it relates to liver risk, or whether it relates to cardiovascular risk. I think we need to know the answer to those questions so that we can best focus on what we say to patients, when and how we say it.
That's an excellent point. A long time ago, Susan Sontag wrote about the metaphors around cancer and why cancer was feared more than diseases that killed more people, because there are metaphors around dying of cancer that are just uglier. And COVID is now that. COVID is the hand of God that comes out of the sky and all of a sudden you're dead. So that metaphorically, that's really powerful. And if you believe that you could get a shield by losing 10% of your body weight, that would be pretty compelling for people who thought they could get it done. We can do this one for a long time, and we're going to do some of it next week, and we'll have other sessions on this later in the year. So why don't we go to final question now? I want to ask each of you two things. Steve, I'm going to give you a slightly different question. For Louise, Ian, and me, the most interesting thing you heard today and the most surprising thing you might come out of in NASH-TAG next month. So surprising for everybody, interesting for the three of us, important for Stephen.
I think the most surprising thing for me, is the diversity of the programming, and that probably reflects the relative successes in development there have been over the last year, both in the diagnostic space and in the therapeutic space. The thing that I think will be most interesting to me is that discussion about whether drugs that are already licensed in diabetes for patients with diabetes and NASH, what impacts they'll have and what additional therapies are really going to be needed. So I'll be really looking forward to that.
I suppose for me, the highlight is the fact that Stephen and the guys from NASH-TAG are probably putting the future real world into a whole morning session with the what does NASH trials look like without histology. So I think that's out there, that's beyond the biopsy. That's hopefully something that I will see in my career as an outcome, I'd like to think, before I eventually retire. But I suppose that for me is the new thing and the exciting thing.
Yeah, surprising for me is the fact that we're close to a sellout for in-person attendance. We were completely unsure if anybody besides the four of us course directors would show up. And we were completely okay with that. But the fact that we are almost at max capacity with 3 weeks to go before the meeting is really a positive sign. I'm excited, tickled to death about that. As far as what's important, you ask a course director, I'm going to say it's all critically important, or we wouldn't have come up with it. But ultimately, I want to see the pulse of the group as it relates to these diabetic meds. What's the feeling? What's the consensus? Can we get a consensus on where that role is likely to be? It's not just GLP-1, it's SGLT2s as well that have impacts on renal disease and heart failure. And then blending that into a co-formulation. Imagine a day where we throw an SGLT2 in with maybe a THR beta or something like that. And where are we with artificial intelligence? We've seen a lot of incredibly good data. We've seen the agency comment that they're excited about how that's going to be incorporated. But what does that look like? How much more data do we need? Can we get consensus from the pathologists on AI as well? I think will be really important. So lots to think about. I think those are my top level thoughts that come to mind about what's important, though.
I look at this agenda, tons of things scream at me. If frankly, if I had one vaccine shot, I would be there. I'm pretty heartsick about not being able to do so. I think the 2 days of diabetes, SEMA day 1, the debate day 2, show some real promise. But having been there for the last 2 years, I saw this experience both times. Somebody says something and demonstrates it, and it becomes blinding flash of obvious for the whole room and changes the scope of what's going on and how people are thinking about lots of different things. I'm confident there'll be a blinding flash for the obvious there. I have not a clue where it's going to come from. And actually, one of the fun things about watching will be to see that. So, Stephen, thanks for walking us through. This has been really a great session. And Ian, Louise, thanks for wonderful questions.
And now, news about surfing Nash.
So that ends today's episode. I want to welcome Stephen back from the cold and the snow. And as Louise points out, he'll get Starbucks back pretty quickly. Thank Louise and Ian for your contributions. Remind everyone that next week includes a topic about treating patients in a world before drugs, using some of the same kind of behavioral issues that Louise was covering this episode, followed by an extra-sode launch of a new integrated non-pharmacological treatment. With that, let me just go on to the business portion of the meeting. As I say, next week is a first for us, in that we'll be introducing a service, and that's exciting. We also will be reporting our plan for NASH-TAG next week, and we're planning for other special events this spring, conferences, sponsors, additions to the ways you can access information from the podcast and elsewhere through Surfing Nash. We can do that because listener growth is continuing. We've averaged well over 2,000 downloads per day as our 30-day average. If you remember when I started talking about 1,000 being a big deal for us, that's a huge number. The last 4 weeks have been responsible for half of our Google page views and a third of our podcast issue downloads after 10.5 months. So I want you to save a couple of dates. Ian will be back on March 29th to talk more about the work that they've been doing in Leeds. He's got 3 papers for publication right now. Hopefully he'll be able to talk about some of that. Alina Allen from the Mayo Clinic will be joining us on April 5th to talk about her paper predicting disease progression in non-cirrhotic and cirrhotic patients based on MRE results. We're working on 4 other special weeks right now. Lots of good things you're going to want to hear, so be sure to stay with us. With that, let me say thank you to my team. Eric Rounds, our social media master, Mike Wilson, who makes us look so good and can tell you where you're going to have some challenges today. Mike Palatea, who makes sure the media all work. Buzzsprout, who did heroic work dealing with their DDoS problem. And most of all to you folks, because you listen and because you send us feedback, we can do more and more interesting things over time. So hopefully people who got real cold and snow and misery last week will do better this week, and no one else will be visited with the same problems. But wherever you are, stay warm, stay safe, surf on. We'll see you on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.